So my name is Nicholas Robert. I'm the Chief Medical Officer for Ontada, trained as a medical oncologist, and I'm very fortunate to have today one of my colleagues, Dr. Paul Conkling, Senior Medical Director. And we're here today to talk about clinical trials and how real-world evidence fits into that process. So both Paul and myself have been involved in taking care of patients for a number of years and also very active in the U.S. Oncology Research Organization, doing clinical trials, running clinical trials. So before we get too far into it, I'd like to ask Paul if he could describe a little bit about the clinical trial process and his perception as being a study PI for some of these protocols.
Yeah, so clinical trials, prospective clinical trials are a mechanism that's used to study new drugs and new treatments, to see how those drugs and therapies work, and also to compare them to standard of care therapies. The typical clinical trials that I was involved with during 33 years of medical oncology practice were primarily prospective trials. And in those trials, the patients that are selected on a very careful basis, so that the patients are all similar in terms of the disease, the extent of disease, and other factors that would influence how they might respond to a treatment. The patients are provide informed consent. They learn about the trial before they decide to enroll. And the role of the PI is really to oversee how that trial process works. The physician, the principal investigator, PI, is the person who often helps to design the trial, to guide the development of the informed consent process, to oversee the trials as it's going on, to review toxicities or side effects that come up, and at the end of the trial, to help interpret the results and look at that information and understand what it really means.
Thank you very much. So what you've heard, this is, prospective, identifying patients, going through a consent process, and as Paul noted, a fair amount of selection. They have to meet certain eligibility criteria. And frankly, we've made a lot of progress in treating cancers this way, going back to childhood leukemia decades ago. And this has been a very effective way of identifying new treatments. It's been realized that, however, that many of the patients, most patients, do not go on clinical trials. And we use clinical trial results to take care of these patients. So this really raises the issue of real-world data and potential of real-world evidence. And frankly, Paul and I probably years ago collected some cases and wrote them up, and that was really real-world evidence. It wasn't given that name. It was collection of case histories. But there's a lot of that in the literature. But the interest in looking at real-world evidence really took off after the 21st Century Cure Act, where the FDA was mandated by this law to begin to look at real-world data and to see how this could be used for real-world evidence. It's a little bit of a different story. Paul, when you're taking care of patients, you use an electronic medical record, and in this case, iNamed. I know you have to do some clicks.
Right.
And you probably, now that you're on the other end trying to make sense of those clicks, you may want to comment on that process about taking care of patients with an EMR.
Yeah, so the way records are kept now in medical care is through an electronic medical record. And part of that information is entered by clicking particular pieces of information, so it's entered in the record for that patient. Some of the information, however, can be put in either by typing or by dictation. And then there are other pieces of information that can be scanned into the chart. Reports, for example, pathology reports or results of scans, x-rays that have been done. And what that does is it results in different kinds of information that you can pull out of the chart. Some of the information can be extracted or removed from the chart pretty easily. Those are the specific pieces of information that have been entered by clicking. Some of the other information takes a little bit more effort to get to. Then you have to really look at the dictated or typed information that's been put into the chart. But all of that information forms the record that is the information for that particular patient.
And so what Paul was describing is what we call real-world data. It's a combination of structured data, those clicks, and unstructured data like the progress notes, radiology reports. And Ontada has spent a lot of time really trying to develop processes that are pretty rigorous so that we have a lot of confidence that the data is extracted is correct. And we go through both verification processes as well as validation processes to ensure that the information we do review that does become real-world evidence, we feel comfortable and can stand behind that data. So there are opportunities to take this real-world data, real-world evidence, and answer certain questions that are particularly relevant. So one of the challenges, Paul, I think, is when you're dealing with a small patient subset, an uncommon cancer or a common cancer, but uncommon because of a particular mutation. And as an investigator doing clinical trials, it can be challenging to do, especially a randomized trial. And can you tell us a little bit about how people do phase 2 trials or single arm trials in this setting, and what are the challenges?
Right. So, Nick, that's a tough nut to crack, so to speak. When you have a group of patients, suppose you have a therapy that is effective for a specific genetic mutation, but this mutation occurs quite infrequently. So there are really only a small number of patients out there with this particular problem. It can be really difficult to find those patients because you're depending on testing being done to find this mutation. In an ideal world, every patient would be tested with a particular disease would be tested for this mutation, but that doesn't always happen. So it is an extra burden on getting a randomized or a prospective trial done in order to find the patients to study. That's the real burden.
So one of the opportunities we have, and this really does require that our data is rigorously collected and analyzed. So Paul could do a study on this population of patients with this uncommon genetic abnormality and treat a patient with a new treatment that he thinks has a promise. And then we can go and find similar patients treated with standard therapies with this genetic mutation, and then compare the results. And we can look at response, we can look at progression-free survival, and we can look at overall survival. And done carefully, we can take the results from Paul's study, his phase 2 study, single arm, and compare it to some of the outcome data that I just described. So that's an opportunity, and one that we'll continue to pursue. This is called, using an external control arm, sometimes called a synthetic arm. Another opportunity is, you get a study and it demonstrates benefit, but maybe not everybody with that disease was tested. So, an example would be patients that are maybe don't have the performance status. And maybe I could ask you a little bit about, so to have people understand the selection criteria. What sort of information is needed to see if a patient's eligible? And maybe you can talk about performance status or even labs.
Right. So typically, you have to be sure that the patient has the proper diagnosis. Very often, trials will want the patient to have a specific stage for the disease, but also the patient needs generally to be fairly healthy other than the underlying disease. That is their performance status, which is really a way of measuring how a patient does day to day, what their activity level is, how they can get around, how they can take care of themselves. Their performance status has to be pretty good to be on a trial. If patients have lots of underlying medical problems, often they can be excluded from a prospective trial. There are also other parameters like kidney function, liver function, blood counts, other laboratory studies that have to be within certain limits for a patient to be eligible for a trial.
So, and you may ask, that's not fair that these patients have to meet a certain criteria to be a participant on a trial. But if you're thinking about it from a life science perspective, there's been a fair amount of research that's been done, investment in terms of trying to identify a new treatment, and you really want to give that new treatment the best chance of doing well. So if you have a patient that's, quote, too sick, we know that the chances of getting some improvement is going to be more difficult. So from a clinical trial perspective, the eligibility criteria that you just heard makes sense. But here we are with real world data. And, Paul has done a randomized trial and he's demonstrated that he has a new treatment that works, but with all that selection criteria that we talked about. And we can now go into our EMR and identify that real world data and analyze it so it becomes real world evidence. And we can loosen up some of that criteria. Maybe, someone with a performance status of, instead of being 0 or 1, a 2, or someone maybe with mildly elevated renal dysfunction, kidney dysfunction. Maybe the blood counts weren't, tippy top, and it could be a little bit compromised, but certainly reasonable to give that treatment. And, we've done that in Ontada. We've looked at, for example, metastatic breast cancer, a population called HER2 positive, and took a treatment that was demonstrated from a randomized trial to be effective. And we looked at a group of patients without those restrictions and demonstrated some benefit. And frankly, it's reassuring, not just as a clinical investigator, which we both were, but taking care of patients, that we can tell our patient that, this treatment was actually given to patients not on a trial and similar benefits were achieved. So I think that I find that useful, knowing that information. Paul, I'm sure you feel the same.
Absolutely. I mean, being able to look at really the same population of patients that I would see in the office and know that this therapy is effective. And because you're looking at so many patients in that kind of retrospective view, that real world view, it's very reassuring that the experience there is something that you can count on for your own patients.
So that's another example of the utility using real world evidence. And I'd like to finish up with one more example. And this is where, the FDA has approved a treatment. It's called accelerated approval process. There is sufficient data, and usually it's in a situation where there is an unmet need, that there is a desire or a requirement to get a better treatment, to find a better treatment, but there isn't all the data that maybe we would like. So this regimen is approved, but with the understanding that the life science company will do more studies. But in the meantime, the drug is available, or the treatment's available. And in that setting, we're able to see how patients do with this new treatment. And again, I'm going to ask Paul, the value of getting that information in terms of as both a researcher and as a oncologist.
Right, that kind of information really verifies the original trial. You can follow patients as they begin this new therapy. You can see a broad range of patients getting the new therapy. You can look for perhaps unexpected side effects that you may not have seen in the previous original trial. And you can look at all of that data together and really get a very realistic view of how the therapy is working, and use that to reassure the FDA that the approval of that drug is appropriate.
So we've gone over some different examples of how we can use real world evidence. And I think many of us today, and I have to say this is an opinion that's changed during my career, that if